Climate change is one of the most pressing issues facing humanity, and it is not a future problem. Changes to the Earth's climate driven by emission of greenhouse gases have led to glaciers shrinking, plant and animal geographic ranges shifting, and historical level droughts, wildfires, and rainfall. But what does all of this have to do with the clinical lab? Let's find out. Welcome to Editors in Conversation. Please subscribe to this podcast and share with your friends and colleagues. This episode is brought to you by the Journal of Clinical Microbiology, available at asm.org/JCM. Editors in Conversation is supported by the American Society for Microbiology, which publishes JCM. If you are a member of ASM, you can get up to 50% off the publication fees of your article when you publish in JCM or any of the ASM journals. I'm JCM Editor-in-Chief Romney Humphries, and today I'm joined by Dr. Joe Wiencek, who is the Director of Clinical Chemistry and an Associate Professor at Vanderbilt University Medical Center. Joe, welcome.
Thanks, Dr. Humphries, and thanks to the Journal for having me to discuss this topic today.
Awesome. Great. And we're also joined by Andrea Prinzi, who is the Director of Scientists for Global Medical Affairs at bioMérieux. Andrea, welcome.
Hi, thanks so much, Dr. Humphries and Joe. Dr. Wiencek, I'm looking forward to talking with both of you about this. It's a cool topic.
Cool. Yeah. Great. Well, I'm excited about this too. So let's dive right in. Anyone who's worked in a clinical lab knows that we are not exactly environmentally friendly. There is a lot of plastics, a lot of waste, and I wanted to start with you, Joe, because I think the bulk of testing that happens across our clinical labs, at least volume-wise, happens in the core lab. So can you talk a little bit about how lab activities impact the environment?
Yeah, absolutely, and glad to touch on this topic. I usually like to start kind of where I kind of influxed into this general area of interest and studies is right around the pandemic. And so walking around, you end up seeing all these, general surgical masks kind of laying around. And so I started to look up some statistics, and there was one that was pretty striking that about close to 9 billion tons of plastic waste were generated during the pandemic from over 190 countries. And 70% of this is associated with hospitals. And then we have, just from that conversation that you had right now, is that a lot of the waste, can be attributed to the clinical laboratory since we're a 24/7 operation. The big thing is that when you dive into the literature, not a lot of studies have been performed. And so we know labs are 24/7, 365 days of the year. And waste is basically present in every single stage of the total testing process. And I kind of like to say that all you have to do is open pretty much any cabinet, drawer, or instrument in the lab to see the waste that's there. Mm-hmm. And one of the last things I'd like to comment as it relates to the core lab is that, yes, we're doing millions of tests per year. And so, this is usually associated with a heavy reliance on single-use plastics. And this is your blood collection devices, your primary tubes, your secondary tubes, tube adapters, biohazard bags. That's a big one that's have come up. And then to top it all off is that our instrumentations, again, run 24/7. And so they're consuming significant amounts of energy and water, adding basically considerably to the lab's overall environmental footprint.
Yeah, absolutely. Joe and I recently moved into a new lab here at Vanderbilt, and we have two humongous generators outside, which are, I guess, exciting from a patient testing continuity perspective, but probably not from the environment's perspective. So yeah, a lot of use. So Joe, you mentioned there's not a lot in the literature for this, and you've actually studied it. Can you tell us a little bit about what you've looked at specifically around basic metabolic panels and the waste generation there?
Yeah, I'm really excited to share this because it was just put into print last month in Clinical Chemistry and Laboratory Medicine, another journal for our subspecialty. And we partnered with 5 other large institutions that had different various analyzers for chemistry. So there's usually a considerable about the big 5 analyzers in total. And the idea was to have a cross comparison of how much solid waste and plastic waste was generated from these 5 analyzer systems. And then we took that information and to kind of like baseline it, we estimated how much waste it would take to run 1 million basic metabolic panels. And so one of the major findings from this was actually an analyzer system that is considered a dry chemistry analyzer, so it doesn't use any water. Ended up having approximately 150,000 kilograms of solid waste that was generated, which was approximately almost 100% plastic.
Wow.
And I know this one. Yeah, a lot of people it's a shock and awe when you see this figure. The other analyzers they ended up putting a lot of solid waste too, but by a factor of 10, the dry chemistry analyzer was by far more out there in the other area of this space, and then on top of it, we ended up expanding one of our original studies, which looked at a comprehensive metabolic panel, but only in one analyzer. We wanted to look at the water and energy usage, but only for one analyzer. So like at Vanderbilt, we have 6 chemistry analyzers. And so we only looked at one just to kind of provide a baseline comparison between these other vendors. And so when we did this for the 5 analyzers of each different vendor, we ended up equating out that those 5 analyzers would take 9, or I guess they would be producing enough carbon emissions as 9 average U.S. homes. So just a huge amount of carbon emissions coming off of very minimal amount of analyzers within a system.
Yeah, those numbers are striking. Yeah, totally. So Andrea, this is a microbiology podcast. So tell me, are we any better in micro? Do we do a better job at this?
No, we don't. Yeah, I think those numbers are so shocking. And I think Core Lab has, obviously, Joe, your work is really impressive. I think they've been able to demonstrate this better than anyone in micro has to date. Mm-hmm. But I think if you think about maybe some of our unique challenges in micro, same issues apply, right? For, labs that are 24/7 and running instruments all day, all night, glass versus plastic, having challenges with recycling. But I think maybe a more unique component to that is that for micro is that single use challenge for us. In micro, we can't risk contamination, right? It has a direct impact on our results and the way patients get managed. And if you've ever lived through, especially as we've moved into more molecular testing, a contamination issue on an assay, it's this huge investigation. And there's a lot that goes into figuring this out and preventing it again. And so it feels maybe a little more restrictive in micro in terms of how creative we can get with reusing or recycling or not doing very strict single use, tactics, if you will, in microbiology. So I think that's maybe the thing that is maybe a little more unique to us. But I also think about just plate usage. Some of the cultures in microbiology are, we're using 10 plates for one culture, or gram staining. If you think about how many gram stains you do a day, if you're just at the bench and you're just doing a quick gram stain off the plate, how much stain are you dumping on a slide? I used to think about that, when I was doing manual gram stains, I'm dumping half a bottle of stain on this slide just because I need to do it fast. And there's so much waste going in this big gram stain reagent bucket. And so definitely micro is we're not, I think micro doesn't tend to be quite as high volume, but I think maybe it's even worse because we're not as high volume and we kick out a lot of waste.
We do. Yeah. Not to mention all the plastics with our molecular tests. So.
Yep.
Yeah. So this is, okay, we painted the picture. We're terrible at this. We need to get better. So I think listeners are probably wondering, okay, this is all good, but what can we actually do to help with this situation? So Joe, let's go back to you. You have a nice paper again on recycling opportunities. And so can you tell us a little bit about that?
Yeah, absolutely. And then going back to Andrea's point too, the fact is you all have a great opportunity to study this. So we've moved in the chemistry space and a bit in the core laboratory, but I think that the avenue is wide open for folks that are interested in this space to look at what can be done in the micro area. And I would encourage it as well. And so, but as it relates to the paper about recycling opportunities, one of the easiest things that I think we could all focus on is going straight to the paper, the cardstock, and the cardboard. These are the most easiest materials to recycle through institutional programs. Once you get into the plastics, there's the universal recycling codes that's usually embedded into the plastics, which is 1 through 6. The higher the number, the more difficult it tends to be of the mixtures of plastics to recycle. So that's a challenge that needs to be basically taken a look at with your group, and then some of the institutional guidelines that might be there. Plus, there's obviously the biohazardous components of it. So if there is contamination, obviously they don't want you to throw that into your recycling areas. One thing I wanted to point out, at least in the core lab, and I think having conversations with our microbiologists here at Vanderbilt, we are basically kind of tied or shackled to our vendors and their decisions. So it's nice to hear, for instance, Andrea and bioMérieux having, just a potential focus on this as a vendor. And so, I'd like to say that, when it comes to this, our research was really aimed at providing this information, one, to let folks that we know that we are interested in this, at least in the United States perspective, because I've heard some individuals say, well, they're in the United States. Why do they care about this? But we care. And so I think it's important to highlight. But one of the things that I think is important for our manufacturers and our vendors is to, call this out during large instrument acquisition discussions. So I would like to say that we should put a sustainability and a plastics usage in all of our requests for proposals in the future, which in my opinion, I think will force competition and drive innovation in this specific space.
Yeah. Yeah. And I think we'll put a pin in that because I know Andrea is going to tell us all about the types of things that they're doing at bioMérieux as an example of the types of initiatives in industry, but absolutely. So Andrea, one angle that I honestly never thought of before, but is really interesting that you've published on is lab stewardship. Most of us are familiar with the idea that you're going to reduce testing from a patient safety perspective or financial perspective. I don't think many of us give too much thought to the potential positive impact of really good lab stewardship programs on the planet. So tell me, what have you found about this?
Yeah, I love this angle. I'm a little biased because I've done a lot of work in laboratory, diagnostic stewardship. But I think, again, no one's really made a very clear link to this, but I think when you think through the literature we have on diagnostic stewardship and you sit there with it, you're like, wow, it's really obvious how much waste we could reduce, right? By just reducing unnecessary testing. And I think when I say it out loud, it seems really obvious, but when you look through some of the data, it's really, I think, pretty impressive. This came to mind for me when I was working on a project with some fabulous people that are doing sustainability. And I think I'll mention them a bit more a little later, but we did this OFID publication on greening in research laboratories, veterinary medicine, and clinical microbiology labs. And, I think to me, diagnostic stewardship feels like the low-hanging fruit because it's getting a lot of attention right now, and there's lots of work happening in stewardship. And I think some good examples are, for example, the Bright Star Initiative by Dr. Woods-Hill and her group, looking at reducing unnecessary blood cultures. I think they were able to reduce their blood culture volume by over 30%. And those were deemed, unnecessary. And if you think about just the actual, reduction in waste just from that alone, but then any subsequent workups that happen from contaminated cultures, anything like that, I think that's really impressive. There's great work out of Johns Hopkins with Dr. Anna Samuels and her group there doing reductions in unnecessary respiratory testing, which respiratory testing is, we do a lot of extra testing there, and we know it drives unnecessary antimicrobial use and all this stuff. But when you look at just the numbers that they were able to reduce, I think it was over 41% of their cultures were reduced. And again, we talk about plates or additional testing that happens on a respiratory culture, all the slides, the plates, any molecular testing, that's a lot of waste with just one culture. So to think that 40% of theirs were deemed unnecessary and reduced, really impressive. I think when you think about urine cultures, it's one of the biggest reasons people seek medical care, there's over 10 million visits annually for that reason, right? And every single urine culture is a cup, a plastic cup, and probably a pipette, and things happening in core lab and things. Yeah. And it's a huge area of focus for diagnostic stewardship because we know that they're heavily overused. We overorder cultures like crazy for urine. And so that's just, really, I think, a huge target area. And then there was another really interesting study in 2023 from the Netherlands where they looked at repeat susceptibility testing. We do that a lot. We have kind of these standing dogma, right? We need to repeat susceptibility testing after so many days to make sure nothing's changed. And they did this really interesting study where they were like, nothing's really changing and we're using all these resources and patient care isn't impacted when we don't do that. So maybe we should change the dialogue around how often we do that. So I just think, just thinking about diagnostic stewardship from that angle, I think is another way to consider the holistic impact of reducing unnecessary testing.
Yeah. I mean, imagine if you could get rid of a couple of your blood culture instruments. I'm not sure how much energy they use, but my guess is it's not small. So, because they are
Not at all.
always
Yeah.
on. So, what about other areas of waste, right? So, this idea of stewardship kind of got me to thinking about streamlining QC through IQCPs or lot sequestering so you don't have to do as much, even streamlining validations or verifications. So, do you think those things are worth looking at, or is that just sort of a drop in the bucket? Maybe Joe, do you want to jump in first?
Yeah, I would love to touch on QC. It was one of the first initial kind of dip my toes into this kind of area of research. And so there was a new product that came out in chemistry, and there's two major vendors for quality control materials that you can use. And primarily they come in little vials of 3 mils where you're supposed to take a pipette and you pipette into a plastic pipette into a plastic sample cup. And these newer materials actually have the ability to go directly onto the automation analyzers, cutting out those two pieces of plastic. And so, it's really interesting when we study this that you could reduce up to 60% of waste just from switching QC material types. And so great other place to start in the core laboratory. And then honestly, I love the idea of lot sequestering if the vendors would allow for it, because you're having one large shipment. And so you're going to be reducing all the shipping materials that potentially would go through, the additional gas or, however delivery method kind of that adds up into that sustainability. So if you can get just enough to run you without having multiple shipments with multiple different packing materials, et cetera, I think that's a really good way to look at this from this kind of perspective as well.
Yeah. Andrea?
Yeah, I agree with all of that. Those are excellent points. I think validations, to your point, Romney, they feel so huge. And I hate to keep picking on AST, but I mean, they feel so large. Sometimes you look at how many panels or cards you're using to just do a validation. And I think this gets back to where I always feel very challenged when I think about this in Clin Micro, that some of the regulatory I don't want to say hoops, the pathway, regulatory steps we have to take is, does this turn into a larger conversation about, do we revisit what we really have to do versus where we could maybe, like you're saying, streamline or look at things differently? I think usually people are a little afraid to explore this because, maybe regulatory practices, they feel like hinder them from exploring what they could streamline or cut down, but there may be opportunity there really. Or maybe we're just not exploring it.
Yeah. Well, I think we're all a little afraid, right? We tend to overdo it, and I think it takes a lot to stick your neck out and say, you know what, this is enough. This is all we're going to do. And I get that apprehension, but I agree. I mean, I think that there's opportunity there. So, Joe, I know another thing that you and I've talked about a lot is minimizing specimen collections and using smaller tubes for blood draws. So how do labs set out on this type of initiative? Because obviously that's good for patient care, fewer sticks, fewer blood loss, but it would reduce plastic use as well, right?
Yeah, I'm glad that you've incorporated this question into the conversation today. I kind of like to start off with just referencing a letter that I came across just probably about 2 years ago, and it was published in the Journal of Thrombosis and Hemostasis. And these authors... Is this
a journal that you read often, Joe?
No, not anymore.
To me, that's a very esoteric
PhD
journal.
Work in coag. Yeah.
Sorry.
Not since my years in coag research, which was PhD another lifetime ago.
That explains it.
But one of the things that was highlighted in this is that there's 25 million liters of blood that are wasted in the Western world that are estimated due to over collection for laboratory testing, which we see quite frequently in the core lab. It sounds like through, again, some of the comments Andrea made, it's not too far from microbiology as well. And so we did just recently, I guess it's 3 weeks since a paper went in print, but we did an investigation to re-examine just a small opportunity, and we used vitamin D. And for folks that are not clinical chemists but are aware of diagnostic stewardship, vitamin D is like the poster child for this area of research. And so the study we looked at basically, which we currently use today at Vanderbilt, a 5 mil tube, we made basically a suggestion if we move to a 3.5 milliliter tube, which anecdotally from the data that we've seen, vitamin D tends to be ordered by itself or maybe with a TSH or free T4 in an outpatient setting, which leaves plenty of blood to run those tests. That basically making that switch could reduce 1 gram of single-use plastic per test ordered, which, it doesn't seem like a lot, not for one specific tube, but as folks know with the vitamin D testing in the core laboratory, you can scale this to thousands of tests per year, and this modest change can yield a significant environmental benefit.
Yeah. Remind us again, Joe, how many core lab tests are done annually? Something like in the 7 million or something like that.
Yeah, we're over probably 4 or 5 million, I think, just at Vanderbilt. And we, you would consider that's like a large operation, but we got like a LabCorp or Quest or ARUP or Mayo, those big four, and other academic labs. They're pushing those same numbers, if not even on larger scales. So
it all adds up. For sure. Yeah. So Andrea, as we've mentioned, you work with bioMérieux, and I know there's a lot of effort, not just from bioMérieux, but other colleagues in industry to think about sustainability goals and reducing their environmental impact as a company. So since you have insight, I'd love to hear, kind of what are the types of things that you guys are thinking about at bioMérieux, what comes up in conversations on this topic?
Yeah, I love that you asked this as well, because it's something that I think I've noticed more in the last maybe 3 to 4 years that I'm really proud of the company for being so committed to and then really vocal about. Like, this is integrated into our goals for like the pillars that we have to achieve. It's a sustainability goal, which I think is awesome. And we're a global company. Like, there's, this makes sense, right? But they have, specific carbon emission reduction targets by 2030. I think the goal is to reduce by more than 50% by 2030 compared to what I think we had in 2019. I know there's a lot of work around vendors and supply chain and thinking about kind of what you mentioned earlier, Joe, like packaging, using more locally sourced items and using just less packaging as if we can, if it's possible. I know, it's hard with these types of materials. I think there's a lot that goes into it, but I think the manufacturer, to your point earlier too, Joe, like they're the ones that would really know, right, what can be transported and shipped and packaged. And then thinking too about just overall footprint. So lots of things around maybe travel and delivery, if we can do sea freight versus air freight, or if there's opportunities to do remote support for customers instead of sending people on site all the time, if that reduces travel, thinking about how people are traveling and why. So there's, I think, lots of opportunities throughout the supply chain, if you will, or the life of a product. And also thinking about, I think there are some initiatives around like managing end of life of instruments and things. Because there's, is there an instrument graveyard somewhere where everything goes? I'm not totally clear. And I know that's one of the objectives that has been mentioned. So it's something that's like a very big goal for the company and that they're really highlighting. So I think that's really, really good to see. And I would also say, Joe, you made a point earlier too. I really encourage people to just talk to their vendors about what's possible or what they're willing to do. Or I think, like you were saying, highlighting some of that when you're working with vendors is a great way to keep the conversation going. And especially if you have vendors or manufacturers that are investing in this kind of improvement, I think it's a really great way to kind of, I don't want to say incentivize people, but like you're saying, kind of encourage people to continue this activity because it's really important. And there's a lot of products and waste out there that I think we could reduce. So we used to do that all the time at my old hospital, right? Like we would talk to our vendors about, if we weren't sure we could recycle something or where did it fall in a category? Because I think all of that kind of stuff is very confusing too. If you're really not good at recycling, if you're not sure, sometimes you need help. You want to do the right thing, but you don't know how to handle these things. And sometimes the manufacturer has some really great insight on their products and what you can do with it. So
yeah,
I think at minimum, one of the things that we highlighted to the vendors was just to get rid of the paper package insert. Like it's 2025. There's 2D virtual barcodes pretty much that can be created by yourself. And so I think that at minimum, that would be a great opportunity. And we actually did recently see a vendor at our main campus lab send out a product notification that they were doing just that and following suit with others. So again, small little change that has lasting impact on other places. So.
And I think bioMérieux does that, right? I don't think we get package inserts anymore with our Vitec panels. So yeah, those things are good. And I think to everyone's point, my experience in industry too, the voice of the customer matters. And so if you let your vendors know that this is something that's important to you, it is something that they will make sure they incorporate in their, agendas and plans. So that's good. Yeah. So I'm going to ask a question about national or international guidelines first, and then we'll get to kind of how people can get involved. So are there any national guidelines, international guidelines on improving sustainability in the lab that people can look at? I know there's, green lab initiatives and there's certain certifications for your labs, but I mean, I don't know. Is there anything available that you are aware of?
Yeah, I could take and comment on this. Back in 2022, I was at a international conference, sipping wonderful espresso, enjoying myself in Brussels, and I'm applauded by the organization that throws up on their screen about all the activities that they've done, and it's the International Federation for Clinical Chemistry and Laboratory Medicine. Popped up basically the 2022 Green Labs Initiative, and so they lay out basically water, energy, waste, chemical, which I think is something that's being highlighted. European and folks over the pond or over the Atlantic tend to be a little bit further ahead of the United States, per typical in this specific space. I have not personally seen any national organizations, at least in lab medicine, that have focused on this topic. I know of a nonprofit called My Green Labs that I've had an opportunity to present with the CEO on just a general annual scientific meeting at ADLM this, I guess, was 2 years ago now. So that's probably the closest that I've seen, but I don't know if Andrea, if you've seen anything in your space.
No, I'm totally unaware of anything, to be honest, though. Yeah. I would defer to Jo.
Yeah. I think that the core lab kind of clinical chemists are a little bit ahead of us in microbiology and getting this going. So just a plug out there for our ASM members, something to think about. So you guys have both been involved in this. This is something that you've thought about. What's the best advice you can give people who are interested in leading an initiative at their lab? What are the challenges? What kind of things do they need to think about going in?
I can jump in first and say first, call Jo.
Yes, that's right. Jo.
Obviously, he's awesome at this, and he can give you some advice. Joe's just really great, though. I think, though, I want to give a quick shout out to some phenomenal women that are doing some really impressive work in this space in infectious diseases, which for Clin Micro, those are our friends, right? Those are our adjacent colleagues. Doctors Shreya Doshi and Preeti Jaggi, at Emory. They head up the sustainability, so it's sustainability and then it ends with ID group, and it's infectious disease clinicians primarily that are doing whatever they can in their space to reduce waste, but often it's involving the laboratory. And I heard Preeti talk at a Ph.D. event that I was speaking at, and she inspired me so much with her sustainability perspective. She's also just an amazing human. And so these women have put together this sustainability group. Yeah. And I think it encompasses all sorts of people across laboratory medicine, veterinary medicine. It's a very One Health kind of group. And that's who I worked with on this OFID publication, but they have regular meetings and they do all sorts of collaborative stuff. And so I would say, honestly, look them up if you're interested in this work in ID. I think they may already have resources or things going that you could look into. So I would say touch base with people that are already doing this. And those are some examples, Jo, and then Shreya and Preeti. And then I would just say, Jo's brought this up multiple times, and I think it's super important. I would just say start really small, because a lot of these initiatives feel really big and actually are very big. Trying to tackle all of them at once is probably impossible. And so, like Jo's mentioned multiple times, you may think it's this little tiny change, but they scale up into this really big impact. And so pick one thing and start there. Make it feel achievable, probably.
Yeah, absolutely. I mean, Jo, you've been really successful at getting trainees to help you out with this work. So, yeah. I mean, maybe that's some more advice.
Oh, there you go.
Yeah, I love the fact that trainees have been taking up kind of the flag with this too. And so, I would say the generation, before us and even before them too, coming up are really interested in this subject matter. It's not just in California or Vermont. It's, it's in other places as well. And, we're in Tennessee and this is very important to me. And so, it's happening in the US. Kind of going back to some of the challenges, know your institution a little bit. I think what I found out is Vanderbilt, the academic side, had a substantially well and plated, sustainability program since the early '90s, and then when they fractured off into a nonprofit for the medical center, it's not as strong, but you just have to navigate it a little bit differently. So know your institution and potential differences between the academic and medical center side was one of the big lessons learned. And I love when I go out and present on this to share a little bit of like a Chinese proverb, which, the best time to plant a tree was 20 years ago. The next best time is now. So you can start now. It's the small things, I think, and it's just finding folks that are like-minded. I actually met Andrea through the pandemic online through there, but I did see her paper cite ours, and so I knew that you were interested in the subject matter. So I was excited to see you as an additional correspondent in this conversation today. So,
ditto, Joe. Lots of respect for the work you're doing. Very good.
Thank you.
Great. So I think the last thing I wanted to talk a bit about, because when I did look through the JCM to find articles on this, we have a lot of stewardship articles, and if people are interested in those, they'll be listed in the show notes. But not a lot of research on, the sustainability side of things. And so if you guys, you've both published on this, what do you think people should be looking at? Like, what are the key problems or items that we need to be studying? Maybe Andrea, do you want to start, and then we'll go to Joe?
I feel like for a lot of the research efforts that I am involved in or seeing, an easy maybe win would be to just probably continue to run with that diagnostic stewardship perspective, because that's probably the work you're already doing if you're in Clin Micro or ID. And just thinking about the extra level of discussion you can build around this, or maybe one other thing you could measure that would demonstrate, how this could be greened up a little, right? I've been surprised. I'm relatively new to this role I'm in currently as Director of Scientists for their Global Research Program. And I'm already really impressed at how many times I'm being asked about including outcomes on this kind of thing for diagnostic impact studies. And I love that, right? Like we talk a lot about when we talk about, sepsis or rapid diagnostics for things like bacterial infections in the blood, we talk about IV to oral switch. And maybe there's an opportunity there where in addition to getting someone home early, we're looking at plastics or the IV itself or the resources and wastes that are generated from someone not going home early or something, and there's probably a layer there that we could add in. And so I think we, as researchers, we should start probably trying to encourage this and think critically about how to include it. I think those outcomes are a little hard for us to define at this point because we're really not always sure how to measure those. But again, we could call Joe and he could guide us on that, So I think even the clinical outcomes research we're doing now, there are some maybe easy layers to add that would help start to build this conversation that don't feel so abstract, like going in and just doing a very specific study on waste in the lab, I think certainly is something we should also be doing. But I think in terms of the clinical outcomes area, I'm working in a lot. I think there's some easy adds that we could do to try to.
Yeah. Any health economic study should include some environmental impact kind of outcome. So yeah, totally.
Agree.
Joe,
agree.
Any thoughts? Any additional thoughts?
I think, Andrea, you're hitting it home. I think the conversation today has really kind of, collated this important topic as it relates to sustainability to something that we've been battling for, decades with diagnostic stewardship. And so just as we routinely highlight costs, I would ask that maybe we all as investigators in this space for diagnostic stewardship add in potentially blood avoidance as well to kind of go back to highlighting that. And then sustainability, fewer specimens collected create these profound downstream effects that I think are really important to connect. And, if we see that these interconnected productions are happening, more or less this hits on to the topic that labs can be more sustainable without compromising that general quality or patient care. And I think that, again, hits home for me. I'm sure for you all that are on too. So.
Yeah, that's great. And of course, if people do any of that work, they should come to JCM with their publications. So just to put that plug out. So thank you both. I think this was a really good conversation. I think it's going to provide some food for thought. I think the thing that for me maybe was most telling is Honestly, none of these things are very difficult. You don't have to reinvent lab medicine just to do a little bit of sustainability work. And so hopefully people feel excited and encouraged and motivated to start doing those things because, as I think we've said many times, it's just little things that add up. So that's fabulous. So thank you both for joining me today. Andrea, thank you.
Thank you so much. This was fun.
Yeah. And Joe, thank you as well.
Absolutely. My pleasure.
Great. Awesome. Well, if you've enjoyed this, please subscribe to the podcast and share with your friends and colleagues. Thank you for listening to Editors in Conversation. If you're interested in papers on lab stewardship, the link to several papers in JCM are in the show notes and at asm.org/EIC. And if you're interested in publishing on this topic in JCM, please do reach out to me. Bye all.